Ever wondered how scientists manage to study the complex, microscopic universe within our cells? The answer lies in a fascinating process known as cell fractionation. It's like taking apart a finely crafted watch to understand the function of each cog and gear. But instead of timepieces, we're talking about cells, the fundamental units of life. To begin with, cell fractionation is a technique used to separate cellular components while preserving individual functions of each component. This technique gives scientists a chance to study and understand the inner workings of cells, paving the way for many medical and scientific breakthroughs. The process starts with homogenization. Picture this: a scientist armed with a homogenizer, which is essentially a microscopic blender, disrupts the cells in a tissue. This action breaks the cell membranes, resulting in the release of the cell contents into a solution. This mixture is known as a homogenate, or a crude extract. Next, the homogenate is subjected to a process known as differential centrifugation. This process involves spinning the homogenate at different speeds and for different durations. The idea is that the heavier components will settle at the bottom first, forming a pellet, while the lighter ones remain suspended in the solution, which we call the supernatant. The supernatant is then removed and spun again at a higher speed. This process is repeated several times, each time retrieving the pellet, which contains different cellular components, sorted by their size and density. The final process is density gradient centrifugation, a more refined method that allows the separation of closely related cellular components. The homogenate is placed in a liquid with a gradient of densities and then spun at high speed. The various cellular components position themselves at the level where their density equals that of the surrounding liquid. Now, this might sound like a lot, but remember, it's about breaking down the complex structure of a cell into its individual parts, so we can understand how each one works. It's about separating the gears of the watch to see what makes it tick. In summary, cell fractionation involves 3 key steps: homogenization, differential centrifugation, and density gradient centrifugation. Homogenization breaks down the cells, releasing their contents. Differential centrifugation, with its varying speeds and durations, separates the cellular components based on size and density. Lastly, density gradient centrifugation fine-tunes this separation, allowing scientists to study the closely related components individually. Through cell fractionation, we're able to unlock the secrets of the cell, propelling our understanding of biology forward. From understanding diseases to developing treatments, this technique plays a crucial role in the world of science.